Hi guys, chapter 12, part 1. Like I mentioned from the previous recording, chapter 12 will go over, will require 2 recordings. We're going to go over part 1 today. And the expectation after part 1 is we should have a better understanding the excitation contraction coupling, meaning how a lower motor neuron will basically induce a skeletal muscle fiber or a skeletal muscle cell to generate tension, meaning it will contract and it will generate tension. But before we get to that, we need to review a little bit about the anatomy of each skeletal muscle fiber. So SKM here stands for skeletal muscle. Fibers means cells, or so we say a skeletal muscle fiber or a skeletal muscle cell. So revisiting the structures, subcellular level, meaning the proteins involved. So cellular and subcellular level. So subcellular, I wrote down here molecular level. So I'm talking about specific myofilaments, specific proteins of muscle tissue. For example, thick and thin filaments. Revisiting that, and also going over what is called a sliding filament model. This is a model, the best model to explain molecular contraction at muscular contraction at the molecular level. So when we say contraction at the molecular level, it's called cross-bridge formation, right? So the sliding filament model relies on this cross-bridge formation, meaning this is a fancy technical way of saying contraction of muscle cells at the molecular level, especially for skeletal muscle fibers and cardiac muscle fibers. Smooth muscle fiber contraction is slightly different, but today we're going to focus on skeletal muscle fiber. So cross-bridge formation within skeletal muscle fibers. The importance of calcium and ATP. And at the end of the lecture, you should know which one is more important, calcium or ATP. Okay? And then the concept of excitation, contraction, coupling, meaning what happens, how is it possible for a neuron to induce a muscle cell to contract? Sorry. So we're going to go over the relationship between a lower motor neuron and a skeletal muscle fiber. So that's the gist for the lecture. Let's get this thing started. So histology, meaning the anatomy at the tissue level. So we start with the structure at the cellular level. The plasma membrane, the cell membrane, or the phospholipid bilayer in muscle, we call it sarcolemma. And we did mention about this from one of the previous lectures. The sarcoplasmic reticulum is the equivalent of a smooth endoplasmic reticulum for other cells. And this sarcoplasmic reticulum in skeletal muscle fibers has a very, very critical role. And that role is the sarcoplasmic reticulum is a storage site and a release site for calcium, right? So the important role of calcium to induce cross-bridge formation, and that calcium comes from the sarcoplasmic reticulum within the skeletal muscle fiber. So you see here, it says axon of motor neuron. It should have been axon of lower motor neuron, connecting the previous chapter to this chapter. Lower motor neuron. This muscle fiber, it should have been skeletal muscle fiber / cell. So you see the plasma membrane is called a sarcolemma here. And the sarcoplasmic reticulum. And within the cell, you have multiple types of protein. One structure, it's called the myofibril. And you can think of a myofibril as a train. And within each train, you have thousands and thousands of cars, right? So this is a myofibril. And within each myofibril, you have the smallest structural and functional unit of muscle. And we call that a sarcomere. So this is one sarcomere right here between neighboring Z line. Okay. That's one. Sarcomere. I'm going to pause the recording a little bit. I'm going to be back, okay? Okay, I'm back. So sarcomere, sarcomere. And the sarcomere is the smallest structural and functional unit for muscles, specifically skeletal muscle and cardiac muscle. And within a myofibril, each myofibril, it contains hundreds to thousands of these sarcomere. And when the muscle contracts, when the skeletal muscle fiber contracts, each sarcomere becomes shorter. The length of each sarcomere decreases so that the entire myofibril also becomes shorter. And the hundreds to thousands of myofibrils within one single skeletal muscle fiber shortens, causing the skeletal muscle fiber itself, the length also decreases. It's not only one cell, yet all fibers within a skeletal muscle as an organ will also decrease in length. So when a skeletal muscle as an organ contracts, the muscle becomes shorter. The length of the skeletal muscle as an organ, the entire organ, its length decreases, right? And that starts at the subcellular level, microscopic level, the sarcomere. And within the sarcomere, each sarcomere, you have thin filaments and you have thick filaments. The thin filaments are built from actin, Both thin and thick filaments are proteins, by the way, because actin is a protein. And the thick filament is made of myosin. And the interaction between actin and myosin, that's what gives rise to contraction that we're going to learn shortly. The interaction between actin and myosin, it's called cross-bridge formation. Okay? And we have also a lot of mitochondria. They are important because, as we learned from aerobic cellular respiration, mitochondria, that's the powerhouse of the cell, generating 95% of the ATP supply for the cell. And because of that, you see the skeletal muscle has lots and lots of mitochondria. Right? So this cell is dedicated for generating contraction. So the entire sarcoplasm is myofibril and mitochondria. So those are the two important, predominant structures that are obvious and they stand out in terms of sheer number is the myofibril and mitochondria. And the third component is the nucleus. And the nucleus, each skeletal muscle fiber contains more than one nucleus. So the skeletal muscle fiber here, they are known as multinucleated and this is from anatomy. You should have been taught about this, right? Multinucleated cell, meaning multiple nuclei. And they are not located randomly. They are not inside, in the middle of the cytoplasm or sarcoplasm. They are being sandwiched between the sarcolemma and the sarcoplasm. So they're right underneath the sarcolemma, right beneath the cell membrane. And they are being scattered throughout the fiber. So multiple nuclei per skeletal muscle fiber. Multi means, multiple, right? Okay, more than one nucleus. And we're going to learn later in the lecture also the importance of the T-tubule. And the relationship between the T-tubule and the sarcoplasmic reticulum, critical for calcium release. And we get to learn about that, the central role of the T-tubule inducing the sarcoplasmic reticulum to release calcium prior to cross-bridge formation. It should be an exciting lecture because we get to apply what we've learned. So most of it is nothing new, really. Maybe one slide toward the end of the recording, but most of today's lecture is applying concept that you previously learned from prior lectures. That's the beauty about this class. We build on knowledge that we acquire and then we get it to the next level. So this is going back, talking about the thick and thin filaments. So the thick filament is made of myosin, thin filaments are made of actin. And what happens is when cross-bridge formation occurs, when the sarcomere becomes shorter, this is important, the thick filaments remain stationary. right? So the thick filaments do not move. Only the thin filaments move. So a sarcomere, like I mentioned, is between one Z line to the next Z line, right? So that's your sarcomere. The smallest structural and functional unit of skeletal muscles and cardiac muscles. And when the sarcomere, due to cross-bridge formation, the sarcomere becomes shorter is because of that, because of the thin filament is moving. It's moving toward the M line. Right? The thick filaments remain stationary, but that blue line right there is moving toward the M line. Every single thin filament is moving toward the M line. On this side also, it's moving toward this M line. Whereas the thick filaments remain stationary. Right? So that's the thin filament, and the thick filament I'm going to do in red, just so you can see. That's the thick filament. They stay in place. They don't move, stationary. Okay, so due to cross-bridge formation, it causes the thin filament to move toward the M line, which is the middle of each sarcomere. By doing this, it will cause the sarcomere to become shorter. Each sarcomere's length decreases due to cross-bridge formation. Like I mentioned, sarcomere, structural and functional units of contraction, smallest structural and functional unit of contraction. Specifically, contraction of skeletal muscle fiber and cardiac muscle fiber. Like I mentioned, smooth muscle is a little bit different. So the importance of the filament, sliding filament model, it explains muscle contraction at the molecular level. So sarcomeres become shorter. Thick filaments are stationary. So you have here your thick filaments. I'm going to highlight it here. So the thick filaments remain stationary. They don't move at all. It is only the thin filament that will move. So the thin filaments are this one, these, they're going to move toward the M line, all of them. On either side. So that's what you see here. The sarcomere before cross-bridge formation and the sarcomere after cross-bridge formation. Same thing here, sarcomere before cross-bridge formation and sarcomere after cross-bridge formation. Right? So you see the length is shorter. This is longer. Right? So at the molecular level, at this level, the sarcomere, we don't say contraction, we say cross-bridge formation, meaning there's a process allowing the myosin to interact with actin, and it causes the thin filament to move toward the M line of each sarcomere, the smack middle of each sarcomere. This also gives rise to I band that you learned from anatomy and the A band. The I band is where you don't have overlapping. It's only thin filaments. Whereas the A band is that there's an overlap between thick and thin filaments, so it looks darker, right? So the A band is due to the A band here is this region. Looks darker because you have an overlap of both short and thick and thin filaments. Whereas the I band over here, you only have thin filaments. There's no overlap, right? That I band is only thin filaments, no overlap. But the A band, so this is I band, this is A band. Now, cross bridge formation. Cross bridge formation means the interaction between myosin from the thick filament with the actin on the thin filament. At rest, when calcium is not present, each actin protein on the thin filament, so each circle is one actin protein, and there's a dark circle on there. That dark circle is the binding site for myosin. So myosin comes in and interact with that darkened circle on the actin. Okay, because of that, we call that the myosin binding site. Right, so this darker circle, myosin binding site, right? The black circle, this circle here. And this is one actin, right? That's one actin protein, okay? So each actin contains one myosin binding site. The problem is at rest, that myosin binding site is occupied by a protein filament by the name of tropomyosin. The tropomyosin is a protein, a filamentous protein, or a protein that looks like a filament, and it occupies all the myosin binding site. So it occludes all the myosin binding site. So in other words, if myosin would have to be here, come in, it cannot interact with actin because its binding site is occupied by tropomyosin. So in order for myosin to bind to actin, it requires calcium. When calcium comes in, calcium interacts with a protein complex, which is made of 3 protein subunits called troponin. This troponin interacts with calcium. And when troponin interacts with calcium, it induces a conformational change. It induces a structural change. And when the troponin interacts with calcium and it causes the structural change, it will also pull with it tropomyosin. So when the troponin component complex changes its shape due to its binding to calcium, it will also trigger a slight movement of tropomyosin sliding out by moving, causing a motion, and that motion, the sliding motion, allows the tropomyosin to basically move away from the myosin binding site. So now we have exposed myosin binding site. Each dark black circle now is available to interact with myosin from the thick filament. So every time that calcium is present, the myosin binding site on the actin are exposed, meaning thick filament, the myosin from the thick filament can come in and induce or kickstart the cross bridge formation. So tropomyosin no longer blocks actin, so calcium troponin interaction leads to troponin no longer blocks actin binding site. Cross bridge formation can occur between myosin and actin. So myosin is from the thick filament, actin is on the thin filament. So what you have here is the G-actin protein. Polymerization make it into a polymer. Double helical actin strand. So this is basically two filaments of actin interacting and making a thin filament, whereas the myosin is shown here. And remarkably, the myosin also has enzymatic activity. So on the head of the myosin, you have two components. You have the actin binding site. So this is where it binds to, that actin binding site binds to the dark circle on the G, on the thin filament. And it also has an enzyme capability. So it has the ATPase site. So this ATPase has different rates. So the fastest one can basically cycle up to 5 cross bridge formation per second. We will talk about this, revisit this concept on the next recording, this part 2 of chapter 12. So the fastest contraction that we can generate is each myosin here can interact with 5 different actin molecule per second. So it grabs one, it releases, it grabs another one, it releases, grabs a third one, release, grabs a fourth one, release, grabs a fifth one, release. Those 5 times, 5 interactions, 5 cross bridge formation occurs every single second. That's the fastest contraction that we can generate in human skeletal muscle tissue. And that's because of the type of myosin heads that the muscle displays. So it can be fast ATPase activity, intermediate ATPase, or slow ATPase activity. We're going to focus on the fast and the slow when we get there. ATPase means it hydrolyzes ATP to harvest energy because it needs energy. The cross bridge formation is an active mechanism. So without ATP, you'll learn later, the myosin and the actin cannot disengage. They're stuck with one another. So once you have that myosin head bound to the actin, once you have the thick filament binding to the thin filament through myosin and actin interactions, cross bridge formation, they cannot disengage from one another. The myosin cannot disengage from the actin unless there's ATP. So when ATP comes in, it induces myosin heads to disengage from the actin and binds to another actin, right? So cross bridge formation here requires calcium and ATP. Calcium allows the interaction between myosin and actin. ATP allows disengaging myosin from actin. And that's why when you run too fast and when your skeletal muscle fibers runs out of ATP, your muscle cramps. The cramping is a reflection of myosin incapable of disengaging from actin since there's no more ATP to help with the disengaging motion. This is also why when somebody dies, the first day, 24 hours, the entire corpse is stiff, and we call that rigor mortis, right? Rigor, R-I-G-O-R, mortis, M-O-R-T-I-S, right? So the entire body corpse is like it's hard like wood. And after one day, after 24 hours, tissues start to break down, and then the body basically becomes, relaxed again. But the first day, it happens every time when somebody dies, the entire body becomes stiff like a plank. And that's due to ATP running out and myosin cannot disengage from actin. So this is the role of ATP. So when ATP, so this is the interaction here. So this is the critical step. So once you have this test happening, when myosin is allowed to bind, form a cross-bridge formation with actin, it cannot disengage, right? And the only way for myosin to leave the actin is ATP shows up. The reason is ATP, myosin head has a higher affinity for ATP than actin. So it releases actin and it binds to ATP. And when it binds to ATP, it will hydrolyze the ATP to sling it again for the energy to attach on the next actin. So the cocking of the myosin head, this is high energy form. And that is required for the next step, going back to the cycle. So 5 back to 1. So it binds. And the power stroke requires energy. And that energy has been harvested from this molecule, ATP. So this power stroke, this is basically that black arrow is moving toward the M line that we mentioned earlier. So this causes the thin filament to move toward the M line. And that is due to the myosin grabbing the actin and pull it toward the M line. The entire time, the thick filament remains stationary. This is stationary. So okay, so during cross bridge formation, the thin filament moves toward the M line. The thick filament remains stationary, remains in place. The thick filament does not move. One more thing. To make this efficient and to also to prevent muscle fatigue or muscle cramping, the concept is called out of phase cross bridge formation. Meaning, if you have, for the sake of simplicity, you have 10 interactions possible. So you have 10 myosin heads with 10 actin. The idea is you don't want to use all 10 at the same time. You might use 3 at a time. So you will get, at this moment, 3 myosin head binds to 3 actin, cross bridge formation, power stroke. Once the power stroke is done, another 3 myosin head that hasn't been used now forms that cross bridge formation. Power stroke, another 3 comes in, power stroke. So you have, out of 10 interactions, you can have now 3 sets of 3s in series. So you can generate a smooth contraction, right? So using the same amount of energy, you basically lengthen the time for cross bridge formation. If you use all available myosin with all available actin, then you would be hard pressed, because you can do it only once and you have to wait until the disengaging and then you bind it again. So you cannot generate a smooth cross bridge formation throughout the available time that you have. So by doing out of phase cross bridge formation, you provide a much more efficiency in terms of using the same energy, yet generating a longer duration for cross bridge formation. So you can generate much more tension over a longer period of time using the same amount of energy. Now moving on to the excitation component. We briefly talked about this during the previous lecture. So action potential comes in through the axon. This is the lower motor neuron, axon of the lower motor neuron, and the axon terminal of the lower motor neuron. So the action potential is propagated on the axon, along the axon. It hits the axon terminal and it cannot be regenerated because the membrane, the cell membrane, the plasma membrane on the axon terminal does not have voltage sensitive sodium channel. But that depolarization will induce, will activate, will open another type of voltage sensitive ion channel. And we call these voltage sensitive calcium channel. And once they open, calcium gets in, this massive calcium influx into the axon terminal will induce the vesicles containing the neurotransmitter we call ACH, acetylcholine, to be released into the neuromuscular junction, right? So I want you to, again, one more time, do not use the word synaptic cleft. That is neuromuscular junction. By simple diffusion, it crosses over the neuromuscular junction. It binds to the ACH receptor, the nicotinic acetylcholine receptor, which is an ion channel. It requires 2 ACH molecules per receptor to open them. And when they open, they allow a massive influx of positive charge ions, sodium and calcium, while allowing potassium to leave. So you have a net accumulation of positive charge inside the inner leaflet of the sarcolemma. So that's a depolarization, and that depolarization signal, we call it EPP, end plate potential. And that end plate potential is always above threshold, inducing the sarcolemma to generate the action potential on the sarcolemma, on the muscle side. The motor end plate is also, should have been learned from anatomy. This motor end plate is the region on the sarcolemma that is directly on the opposite side of the axon terminal. That sarcolemma, that region, has a high density of these ACH receptors. We call that motor end plate. And once that action potential is generated, it will be spread throughout the entire muscle fiber, skeletal muscle fiber. And it spreads also deep inside because of these invagination that we call T-tubule. So the invagination of the sarcolemma inside the cell, that structure we call it T-tubule. And once the T-tubule, so the action potential propagates along the sarcolemma into the T-tubule. The T-tubule, the sarcolemma along the T-tubule sits next to the sarcoplasmic reticulum. And we're going to learn very shortly, there are transmembrane protein allowing the interaction, allowing the sarcolemma to detect incoming action potentials on the T-tubule, on the sarcolemma of the T-tubule. And it recognizes that signal and it releases calcium into the sarcoplasm that you see here. And when calcium arrives, it interacts with troponin, inducing a shift of the tropomyosin, exposing the myosin binding site on the actin, allowing myosin to come in to induce cross-bridge formation, power stroke, pulling the thin filament toward the M line, and the process restart. As soon as the action potential goes away, calcium is sequestered back into the sarcoplasmic reticulum, meaning the sarcoplasmic reticulum takes back calcium. When there's no more calcium, troponin no longer interacts with calcium, goes back to its resting state, bring it with it, tropomyosin. So the tropomyosin now sits on top of each myosin binding site on the actin. So myosin can no longer interact with actin, the muscle relaxes. So NMJ here, that's for the neuromuscular junction right here. So instead of synaptic cleft, you call it neuromuscular junction. So this is the detail again. So this is from the previous recording. Sarcolemma is depolarized. So the action potential arriving on the axon of the lower motor neuron depolarizes the cell membrane, the plasma membrane of the axon terminal, activating the voltage sensitive calcium channel. Calcium gets in, inducing the vesicles containing the ACH neurotransmitter to fuse with the membrane of the axon terminal, emptying its content, their content, into the neuromuscular junction. ACH binds to the nicotinic cholinergic receptor, which is an ion channel, allowing sodium influx, allowing calcium influx, and allowing potassium efflux. And so here, there should be another arrow going in for calcium. So you have a net accumulation of positive charge on the inner leaflet of the sarcolemma. And this gives rise to an EPP, end plate potential, that induces the formation of an action potential on the muscle. And once ACH is done its job, it would be killed, neutralized by an enzyme, acetylcholine esterase, cut it into acetic acid and choline. Choline is recycled back to make more ACH. That action potential spreads throughout the muscle, going into the T-tubule, recognized by the sarcoplasmic reticulum, and the sarcoplasmic reticulum releases calcium into the sarcoplasm. I'm repeating on purpose. So the best way to remember and to learn is through repetition, right? So the sarcolemma depolarization generates EPP, end plate potential. End plate potential is always above threshold potential. So the muscle, the skeletal muscle, will always be able to generate that action potential, because on the sarcolemma of the muscle, for it to be able to generate action potential, it must have voltage sensitive sodium channel. It must also have voltage sensitive potassium channel. And it also must have the sodium potassium ATPase to clean up the mess caused by voltage sensitive sodium and voltage sensitive potassium channel. Right? That was one of the questions from exam 2, because when voltage sensitive sodium channel opens, sodium gets in, so you have a massive sodium inside, which is not normal, and you have a massive potassium on the outside, which is not normal either. So this ATPase will kick out sodium and will bring in potassium, restoring homeostasis by using energy, right? So the action potential, the voltage sensitive potassium channel, reestablish the polarity. We call it electrical chemical gradient. So the voltage sensitive potassium channel restores the electrical polarity, homeostasis, but the chemical homeostasis has not been restored. Sodium is still inside, potassium is still outside. That needs to be restored. And that is done by the sodium potassium ATPase. And so when we talk about the action potential, so the depolarization causes a mess because you're flipping the polarity, but the stage 2, the repolarization, restored that polarity. So it makes the inside negative again. But that's just the electrical component. The chemical component here is still a mess. And that ATPase solves the chemical component, right? So electrical, chemical gradient. So voltage sensitive sodium channel reverses the polarity, depolarizes the membrane. That needs to be restored. So that's the electrical component, and that is restored by the voltage sensitive potassium channel through phase 2. But even when phase 2 is done, when the action potential is done, everything is done, we have a problem. Potassium is outside, sodium is inside. We need to get rid of that. And this is exactly when sodium and potassium ATPase comes in to cleanse this mess, to restore the chemical homeostasis. It will bring potassium in, it will kick out sodium at the expense of energy. So it is using energy to clean up the chemical mess left behind by the birth of an action potential. And that was a critical thinking question from exam 2. So end plate potential is acting like a spark plug, if you will, to induce the muscle to generate its own action potential. So we have, it's the same concept, nothing is new, igniting the generation of APs on sarcolemma, because the sarcolemma has its own voltage sensitive sodium channels, voltage sensitive potassium channels, right? So VS Na plus CS stands for voltage sensitive sodium channels. VSK plus CS stands for voltage sensitive potassium channels. And they are on the sarcolemma. That's why they can generate an action potential. And you see it here, the action potential generated and is pressed throughout over here, this direction, down here, and keep going throughout the entire muscle. So it is spreading along the sarcolemma, okay? Because the sarcolemma has voltage sensitive sodium and voltage sensitive potassium channel. Now, when that action potential is spreading on the sarcolemma, goes into the T-tubule here, and there are two transmembrane protein. They are critical. On the membrane, the sarcolemma region that we call T-tubule, it contains the DHP receptor. Okay, so DHP receptor, that's on the T-tubule. T, which is the sarcolemma, by the way. In the invagination of the sarcolemma, we call it, the T-tubule is the structure, that the space that causes the invagination of that cell membrane of the skeletal muscle fiber. The other transmembrane protein, the ryanodine receptor, which is on the membrane of the SR, the sarcoplasmic reticulum. And the two transmembrane protein physically comes into contact with one another. So when the action potential spreads through the sarcolemma in the region called T-tubule, it will induce a conformational change, a shape change on the DHP receptor. And that conformational change, or the change in conformation of the DHP, conformation here spells CONFORMA, T-I-O-N, conformation. This conformational change will also induce a change on the ryanodine receptor. It's like a lid, so it opens a lid of a can, so the calcium, this is inside SR, will leave, and this here goes into the sarcoplasm. So this calcium now goes into the sarcoplasm. Right. Whereas here, this side, this is inside the sarcoplasmic, oops, inside SR, inside this one. And the space here and here, that's your sarcoplasm. So that's the deal. Every time an action potential propagates throughout the skeletal muscle fiber, every time the action potential propagates throughout the entire sarcolemma, including when it propagates along the sarcolemma that invaginates the cell, that structure, that region, we call it T-tubule, it will be detected by transmembrane protein that are sensitive to the arrival of this depolarization signal. So these DHP receptor are sensitive to depolarization. It induces the DHP to change its shape. And when the shape changes, it will also cause a shape change on another interacting protein called ryanodine receptor, which is the transmembrane protein that is located on the membrane of the sarcoplasmic reticulum, because the sarcoplasmic reticulum is a membranous organelle. So once you basically change its shape, it opens up, and then there's a pore, calcium just sneaks out, and that's an example of a facilitated diffusion. Calcium just diffuses out based on concentration gradient. And when calcium gets out, you know the rest is history. Calcium interacts with troponin, inducing a shape change, bringing with it sliding the tropomyosin away from the myosin binding site on the actin. Now the myosin binding site on actin is available for myosin to come in for cross-bridge formation. And in the presence of ATP, we have cross-bridge formation, moving the thin filament toward the M line, shortening sarcomeres, which leads to the shortening of the myofibril, which leads to the shortening of the entire skeletal muscle fiber, leading to the shortening of the entire skeletal muscle as an organ. We have muscle contraction. So on that note, I'm going to end today's recording, and then we should be continuing on the next recording, part 2. Until then, be safe out there. Take good care of yourself. Study well. Take care.